Visceral fat will kill you. Not the fat you can pinch, not the fat you can see. The fat wrapped around your liver, your pancreas, your intestines, and the base of your heart. Invisible, undetectable without imaging. And according to research published in the New England Journal of Medicine, a more powerful predictor of cardiovascular death and cancer development than your body weight, your BMI, or the fat you can actually feel. Here is what no one is telling you about visceral fat. It is not simply stored energy. It is a metabolically active tissue that secretes pro-inflammatory cytokines, including IL-6 and TNF-alpha 24 hours a day. The same cytokines that suppress your immune system's ability to identify and destroy cancer cells before they become malignant. Visceral fat does not just increase cancer risk by carrying extra weight. It actively dismantles the immune defense designed to catch cancer before it starts. And there is one common vegetable, one that costs less than $1 per pound, sits in every supermarket produce aisle, and has been scientifically documented to directly trigger the programmed death of visceral fat cells and simultaneously reactivate the branch of the immune system that cancer cells most fear that almost nobody is eating. I am going to show you exactly what it does, why it works, and the one mistake in how most people prepare it that eliminates the primary compound responsible for everything I just described. Every claim in this video is sourced from peer-reviewed journals, not here to sell you a supplement, here to give you the evidence. The vegetable is red cabbage, purple cabbage, the same vegetable. The dark-leafed, densely folded, deep violet head that has been sitting in your supermarket produce aisle for your entire life, positioned directly next to the kale and spinach you are probably already buying, and that almost no health-conscious person is eating regularly. It costs less than $1 per pound. It lasts 2 weeks in the refrigerator, and it contains a combination of compounds that, according to research published in 4 separate peer-reviewed journals, directly trigger visceral fat cell death, reactivate natural killer cells, your immune system's primary cancer surveillance units, and produce a biological effect that not even the most expensive supplement protocol has been able to replicate. Here is what those compounds are and what they do. The deep purple-red pigment in red cabbage is not a side effect of its biology. It is one of the most biologically active compounds in the human diet. These pigments are called anthocyanins, a subclass of flavonoids responsible for the red, purple, and blue colors of many plants. And red cabbage contains one of the highest concentrations of dietary anthocyanins of any commonly available vegetable. Here is the mechanism, and this is the part I want you to understand clearly before we continue. Visceral fat cells, adipocytes surrounding your abdominal organs, do not simply accumulate until something external destroys them. Under the right biological conditions, they self-destruct. This process is called adipocyte apoptosis, programmed fat cell death, and anthocyanins are among the most potent dietary inducers of this process identified in the research literature. A clinical trial published in the European Journal of Nutrition administered concentrated anthocyanin supplementation to overweight adults over 12 weeks and measured visceral fat directly using MRI imaging. Not body weight, not waist circumference, but actual visceral adipose tissue volume. The result, statistically significant reductions in visceral fat volume in the anthocyanin group compared to placebo through a mechanism specifically identified as downregulation of adipogenic gene expression, the genes responsible for forming and maintaining fat cells. The mechanism at the cellular level involves 2 pathways. First, anthocyanins activate AMPK, the same energy-sensing enzyme activated by cold exposure and exercise, which shifts cell metabolism away from fat storage and toward fat oxidation. Second, and more specifically, They suppress PPAR-gamma, the master transcription factor for adipogenesis. When PPAR-gamma is suppressed, visceral fat cells cannot maintain their size and number. They undergo apoptosis. They die. The fat does not migrate. It is biologically eliminated. The cancer connection is direct. Visceral adipose tissue is not passive. It actively secretes interleukin-6 and tumor necrosis factor alpha, both of which suppress NK cell activity and promote the tumor-permissive inflammatory microenvironment. Reducing visceral fat load through anthocyanin-mediated adipocyte apoptosis measurably reduces this inflammatory output and measurably restores immune function. Eating red cabbage daily begins this process. The anthocyanin concentration in 2 cups of raw red cabbage is sufficient, based on the dosing parameters of the clinical studies, to begin inducing AMPK activation and PPAR gamma suppression within 72 hours. Stop for one moment. What I just described, anthocyanins killing visceral fat cells, is the most well-documented mechanism. But it is not the most surprising thing about this vegetable. The second mechanism I am about to describe involves a compound that directly activates the specific immune cells responsible for identifying cancer cells and eliminating them before they can replicate. Research published in the Journal of Nutritional Biochemistry found that this compound increased natural killer cell activity by measurable significant amounts in human subjects without a pharmaceutical, without a supplement, through diet alone. And the third mechanism, the one that will most directly affect how you behave in your kitchen starting tonight, involves the way you are almost certainly preparing this vegetable right now. A preparation method that destroys the primary immune activating compound completely before it ever reaches your bloodstream. Stay through all 4 mechanisms. The fourth one, the fermentation finding, is from a Stanford randomized controlled trial published in Cell. It is the most counterintuitive piece of data I have reviewed. Red cabbage belongs to the cruciferous vegetable family, the same family as broccoli. And like all cruciferous vegetables, it contains glucosinolates, sulfur-containing compounds that are converted upon chewing or cutting into biologically active molecules with documented anti-cancer properties. The specific glucosinolate most relevant to immune function in red cabbage is glucobrasicin. When converted by the enzyme myrosinase during chewing, glucobrasicin becomes indole-3-carbinol, a compound that interacts directly with a receptor inside immune cells called the aryl hydrocarbon receptor, or AHR. AHR activation by indole-3-carbinol does something specific and important. It promotes the differentiation and activation of natural killer cells, the immune cells that patrol the body for cancer cells, virus-infected cells, and cellular debris with abnormal surface markers. NK cells are sometimes called the immune system's first responders. They do not require prior exposure to a specific antigen. They recognize and destroy abnormal cells immediately without preparation. They are the biological system most directly responsible for identifying cancer cells before they can establish a tumor. Research published in the Journal of Nutritional Biochemistry demonstrated that indole-3-carbinol supplementation significantly enhanced NK cell cytotoxicity, the ability of NK cells to destroy target cells in human subjects. A separate study in the journal Nutrients found that cruciferous vegetable intake was independently associated with higher NK cell activity in a population study of more than 1,000 adults. There is a second immune mechanism specific to indole-3-carbinol that is equally important. It inhibits the aromatase enzyme reducing estrogen production in adipose tissue. Excess estrogen produced in visceral fat is a well-documented driver of estrogen receptor-positive breast cancer and endometrial cancer. Reducing local estrogen production through dietary indole-3 carbinol is a cancer prevention mechanism that operates entirely independently of the NK cell pathway. Two separate anti-cancer effects from the same compound. Eating 2 cups of raw red cabbage daily provides approximately 100 to 200 milliliters of glucobrasicin precursor within the range shown in clinical research to produce measurable NK cell activation effects. Before we discuss preparation, the thing that determines whether either of the compounds I just described actually reaches your bloodstream, I want to tell you what happens to most people who hear this information. They go home, they buy red cabbage, they cook it, and in doing so, they eliminate the primary mechanism responsible for everything I just described. Mechanism 3 is the one that will most directly affect what you do tonight. And Mechanism 4, the fermentation finding from Stanford, is the intervention that multiplies the immune effect of this vegetable by an order of magnitude that most people find difficult to believe. If you eat cruciferous vegetables, if you buy broccoli, cabbage, Brussels sprouts, cauliflower, there is a very high probability that you cook them, you roast them, steam them, sauté them, add them to soups, or stir-fries. You do this because you have been told these are healthy foods, and cooking them makes them more palatable. Here is what you have not been told. Myrosinase, the enzyme that converts glucobrasicin into indole-3 carbinol and glucoraphanin into sulforaphane, is completely inactivated at temperatures above 70 degrees C. This is well below boiling temperature. It is well below roasting temperature. It is below even gentle steaming temperature if held for more than 3 or 4 minutes. When you cook cruciferous vegetables, including red cabbage, you destroy myrosinase before it can perform the conversion. The glucosinolates remain in the food. They enter your digestive system, and they are partially converted by gut bacteria at a rate that research has shown to be 60 to 90% less efficient than the myrosinase-driven conversion that occurs when the vegetable is consumed raw. This means that the cooked red cabbage, the cooked broccoli, the roasted Brussels sprouts that millions of health-conscious people eat faithfully as part of their cancer preventive diet are delivering a fraction of the indole-3 carbinol and sulforaphane that the same vegetables would deliver raw. I want to be precise. Cooking does not make cruciferous vegetables harmful. They retain fiber, vitamins, minerals, and many beneficial compounds. But for the specific immune-activating and cancer-preventive mechanisms we have discussed today, the NK cell activation and aromatase inhibition driven by indole-3 carbinol, raw preparation is not optional. It is the mechanism. There is one exception that does not involve heat, and it produces a result that makes raw cabbage look modest by comparison. That is Mechanism 4. What to do starting tonight? Add 2 cups of raw red cabbage shredded or thinly sliced to a salad, a wrap, or any cold preparation. Chew it thoroughly. Chewing is the physical mechanism that ruptures cell walls and allows myrosinase to contact glucosinolates. A brief massage with lemon juice or apple cider vinegar, both acidic, both myrosinase preserving, enhances cell wall breakdown without heat damage. This takes 90 seconds of preparation. What I am about to describe is why I specifically chose red cabbage for this video above every other cruciferous vegetable. Not because of its raw glucosinolate content, not because of its anthocyanin concentration, but because of what happens to it when it ferments. And what the Stanford research team found in a randomized control trial published in the journal Cell in 2021 is, in my view, one of the most significant findings in nutritional immunology in the past decade. Sauerkraut, kimchi, fermented red cabbage, these are not exotic foods. Sauerkraut is available in every supermarket. Kimchi, fermented Napa cabbage and vegetables, has been consumed across East Asia for centuries and is now widely available. Both are produced through lactofermentation, the natural process by which lactobacillus bacteria on the surface of cabbage convert sugars into lactic acid, creating an acidic anaerobic environment in which the bacteria multiply to extraordinary concentrations. A single tablespoon of properly fermented sauerkraut or kimchi contains approximately 1 billion colony-forming units of live lactobacillus bacteria. 1 tablespoon. Now here is the Stanford finding. In 2021, a team at Stanford School of Medicine, led by Dr. Justin Sonnenberg and Dr. Christopher Gardner, published a randomized control trial in the journal Cell, comparing 2 dietary interventions over 10 weeks, a high-fiber diet versus a high-fermented food diet. The fermented food group consumed sauerkraut, kimchi, kefir, kombucha, and fermented cottage cheese, foods readily available in most supermarkets. The findings were striking enough that they generated commentary in multiple major scientific journals. The fermented food group showed a statistically significant increase in microbiome diversity. 19 distinct microbial species increased. They showed a significant reduction in 19 markers of systemic inflammation, including key cytokines involved in immune suppression, and they showed measurable increases in the activity of immune cells, specifically a significant upregulation of the immune cell populations responsible for pathogen and cancer cell surveillance. The high-fiber group showed no comparable immune improvement. In fact, fiber alone did not significantly reduce inflammatory markers. Fermented food did. The mechanism connects directly to everything we have discussed today. Lactobacillus bacterium in fermented cabbage produce short-chained fatty acids, particularly butyrate in the colon. Butyrate is the primary fuel for the cells lining the intestinal wall, and it directly supports the integrity of the intestinal barrier. When the intestinal barrier is compromised, a condition called intestinal permeability or leaky gut, bacterial components enter the bloodstream and trigger systemic inflammation. The same chronic low-grade inflammation that suppresses NK cells and enables cancer cell survival. Restoring gut microbiome health through fermented cabbage consumption reduces this intestinal permeability, reduces systemic inflammatory signaling, and removes one of the primary suppressors of the NK cell activity that indole 3-carbinol is simultaneously trying to activate. Raw red cabbage activates NK cells through indole 3-carbinol. Fermented red cabbage removes the inflammatory environment suppressing those same NK cells. They are not alternatives. They are complementary, and together they address immune function from 2 separate biological directions. What to do? Add 2 tablespoons of raw sauerkraut or kimchi to 1 meal per day. It must be raw, refrigerated, and labeled live cultures or naturally fermented. The shelf-stable pasteurized versions have had all live bacteria eliminated through heat. Refrigerated section only. 2 tablespoons is the minimum serving associated with meaningful microbiome effects in the research literature. Let me offer a perspective on what we have covered today. Red cabbage, 1 vegetable, 4 mechanisms, anthocyanin-mediated visceral fat apoptosis, indole 3-carbinol-driven NK cell reactivation, the preparation method that determines whether either of those mechanisms occurs, and fermentation that multiplies and protects the immune effect at the gut level. This is not exotic information. It is not cutting-edge laboratory research from a study that has not yet been replicated. The anthocyanin visceral fat research has been published and replicated. The indole 3-carbinol NK cell literature spans more than a decade. The myrosinase heat sensitivity data appears in basic food science textbooks. The Stanford fermented food trial was published in one of the most respected scientific journals in the world, and yet most people who are making deliberate, careful dietary choices for their long-term health People who spend money on supplements and organic produce and specialty health foods walk past red cabbage every week without giving it a second thought. The gap between what the science knows and what actually reaches people making food decisions is still enormous. Closing that gap, one video at a time, is why this exists. 3 things starting today. Buy a head of red cabbage. Shred 2 cups. Eat it raw in a salad, a slaw, a wrap with lemon juice every day. Do not cook it if the goal is indole 3-carbinol and myrosinase-driven conversion. Cooking eliminates the primary mechanism. Add 2 tablespoons of refrigerated live culture sauerkraut or kimchi to one meal daily. Refrigerated section. Live cultures on the label, not shelf-stable. Do both together. The raw cabbage activates NK cells. The fermented cabbage protects the immune environment those NK cells operate in. Together, they address immune function from 2 directions simultaneously, at a combined cost that is less than the cheapest probiotic supplement on the market. Which mechanism surprised you most? Was it the anthocyanins and the finding that a pigment gives red cabbage its color, while simultaneously triggering the programmed death of visceral fat cells through AMPK activation and PPAR gamma suppression? Was it the NK cell activation and the research showing that dietary indole 3-carbinol measurably increases natural killer cell cytotoxicity in human subjects without a supplement? Was it the preparation finding and the discovery that the cooking method used by most people who eat cruciferous vegetables eliminates 60 to 90% of the primary immune-activating compound? Or was it the Stanford fermented food trial and the finding that 2 tablespoons of sauerkraut per day outperformed a high-fiber diet in reducing inflammatory markers and improving immune cell activity in a randomized controlled trial? Tell me in the comments. And if you have been eating red cabbage, raw or fermented, tell me that too. Your experience helps shape what we cover next. Think about someone specific. Think about someone in your life who is health conscious, who reads labels, who buys organic, who takes supplements, who has deliberately organized their diet around anti-inflammatory and immune-supporting principles, who walks past a head of red cabbage every single week at the grocery store without a second thought, because it has never been positioned as the thing they are looking for. Think about someone managing their weight, who has tried caloric restriction, who has tried exercise, who has not been told that visceral fat is not simply a caloric excess problem, but an active inflammatory tissue that secretes cancer-promoting cytokines, and that there is a $1 per pound vegetable that directly induces visceral fat cell apoptosis through a dietary mechanism. Share this video with them, not to lecture, to give them the same access to the same evidence you now have. Because what you do in your kitchen, specifically which vegetables you buy and how you prepare them, has measurable biological consequences. And the research supporting those consequences is published, replicated, and accessible. It simply has not reached most of the people whose daily decisions it should be informing. Everything in this video is for educational and informational purposes only. It does not constitute medical advice, a clinical diagnosis, or a treatment or prevention recommendation for any specific condition, including cancer, cardiovascular disease, or metabolic disorders. The findings described reflect peer-reviewed scientific literature available at time of production. Individual responses to dietary changes vary. Consult your physician or a qualified healthcare professional before making significant dietary changes, particularly if you have an existing medical condition or are managing weight-related health issues. The goal is to translate published research into accessible information, not to replace personalized clinical guidance. If this story changes the way you look at the common vegetables in your grocery aisle, please subscribe and like the channel. We are unearthing the science that often remains buried in journals. Plant the seeds for your future health. The next treasure of nutritional evidence will be opened soon.